Well, good morning or good afternoon, everybody, and happy first day of meteorological spring. And welcome to yet another fall publishing webinar. I'm Chris Beatties, editor of Growertalks and Green Profit Magazine, and I'm going to be your host today for the next hour or so as we tackle the topic, well, it's right there on your screen, managing ethylene risk in the supply chain. Now, this is a pretty fascinating topic. I've learned a lot going through the rehearsals. One thing I think you're really going to walk away with is a much better understanding of how common ethylene damage is and yet how often it's undiagnosed or even completely ignored. Now, as always, I am not the expert on the topic, but I've got not just one person who is, not just two, not three, but I've got four experts today, and in fact, it takes two hosts to keep them all in line. So let's dig into who they are. First, we have got, well, you know this guy because I've done webinars with him. This is Dr. Vijay Rapaka, the Corporate Grower Research Manager for Oasis. Vijay, are you there, my friend?
Yes, he is.
He's there. Well, that's right. We've got three people in one room sharing a mic, so I'll save those intros for later. Next. Next is Dr. Anil Ranwala, Chief Scientist, and he looks like it there in that lab coat from Floralife. Anil, are you there? You're broadcasting from another location.
Yes, Chris, I'm here. Good afternoon.
You're down in South Carolina. I'll tell you everybody where our experts and our hosts are in a second here. Next comes Sanal Krishnan, also a doctor. Welcome, Sanal.
Hi, Chris. How are you doing?
Very well, thank you. And he's not a doctor, but he plays one in this webinar. He is Tanouye, General Manager, so he's an important guy for the Americas with Floralife. Hi there, Alan.
Hey, Chris.
And last but hardly least, my co-host, Mr. Bill Riffey, the General Manager, Grower Operations for the Americas, Europe, and Africa. I'm surprised, Bill, you had a chance to sit down long enough to help me out with this webinar.
It's great to be here, and I'm not sure exactly where I'm going next, but I'm at least in Kent, Ohio today.
Kent, Ohio. So that's where you and, let me get this straight, Vijay and Sanal are all sitting there side by side, sharing a microphone and supposedly a set of headphones. They're at the general headquarters for Floralife, correct?
Well, the general headquarters for Smithers-Oasis, for the whole world, actually, right here in little Kent, Ohio.
Gotcha. All right. And Vijay, you and Sanal are both broadcasting from your own offices in South Carolina, right?
No, they're actually right here sitting beside me.
No, I'm sorry. Anil and Alan. I've got notes all over the place. I told you we got too many people here. I'm sorry about that. Anil and Alan, you guys are in South Carolina.
Yes.
Yeah, that's right.
There we go. Okay, and everybody is connected, which is the beauty of webinars. You can do them from pretty much anywhere on the planet as long as you've got an internet connection. And me, well, you know me, I'm broadcasting from the usual spot, so we're not going to waste any time talking about that. I will tell you I'm trying a new computer system this time, so hopefully that will eliminate some of those garbled audio issues we've had. If you've been on my webinars before, you know it at about 12:50 Central, suddenly I start sounding like I've been drinking or something. I swear I haven't. It was the computer. I promise you. Anyway, let's handle a little bit of housekeeping here. The first thing being, if you can't hear me right now, and amazingly enough, I haven't gotten a single message from someone saying, where's the audio? So that is a beautiful thing. If anybody can't hear, this is what you need to do. Click on the proper messages there at the top. If you have questions as we go along, there's two areas where you can put them in. You can put them in the chat area or in the Q&A area. Your choice. Just pick your favorite. That'll work fine. We'll get to them as we go along, and if they're appropriate to the topic at hand. And if not, we'll save them for the end. We'll leave 10 minutes for Q&A. And if we don't get to your question, or if you have something kind of complicated, I'm going to give you an email where you can send your questions in. So how about that? And if for some reason either you have to leave the webinar or you want to share it with a friend or watch it again, we're going to archive it as soon as possible afterwards. And to the fellow who said he doesn't have a microphone, good. I've got too many speakers as it is. So that's it. I think that's pretty much all of my housekeeping. Bill, I am going to turn it over to you as my special guest co-host to handle the both the kind of the intro to the topic, and then you can handle the intro to each of our experts as they cover their respective topics. All set?
I think we are. I'm just hoping I can fill the shoes of being a co-host here.
It's a tough job, but at least you don't have to push any buttons. I'll take care of that. So,
okay.
Take it away.
Well, why don't we jump into this and managing ethylene risk in the supply chain. And you're right, Chris, like you mentioned earlier in the conversation, it's everywhere. And we're going to go through and show really how prevalent it is and how to mitigate some of the risks. So if you think about it, and if you want to get to the next slide here, and I'm probably going to get in trouble from Geico because I didn't ask them permission, so this is a free ad for them. We think about the supply chain, and ethylene is an accident waiting to happen. It's everywhere around us. There's other drivers and things that you can't see. And a lot of what we need to do is provide insurance through the supply chain to make sure that if there are problems, you are protected. And I think that's kind of a theme of looking at ethylene, because ethylene, you'll see from Anil's presentation and the damage it could cause. I think it's misdiagnosed. Sometimes it causes secondary damage, like in petunia meltdown. And all these different factors affect the quality to the consumer and your sell-through. And it's a real shame that you've worked all season and you just shipped an entire truckload of bedding plants to Home Depot or to local garden center, and it comes off and there's not one flower in the plant. So these are things that we're going to talk about, mitigating that risk and identifying when you do have a problem, but a lot of how could you stop this going forward. So I'm going to move over to Alan. You want to take it away and talk about some of the work we've been doing for the last 20 years from the standpoint of ethylene-sensitive crops and stems in the cut flower side, because this is where it started. And I think you'll kind of can give a scope of how big a problem ethylene is in the ornamental industry and also in the cut flower industry. So Alan, I'll turn it over to you.
Okay, Bill. Yeah, so I agree. Insurance is a good metaphor for how we think about ethylene control or ethylene mitigation. And I think on the cut flower side of the industry, we recognized ethylene as a problem for quite a while. I mean, it started with STS treatments at first, silver thiosulfate, and then in about 2000 with Ethylbloc. And we've been very fortunate to work with so many great customers on ethylene problems, and we've been able to see so many situations as a result. But of course, not all flowers are ethylene sensitive. So if you could go back a slide, right. We think that by our estimate, about 6 out of 10, or about 60% of all flowers sold in the United States today, I'm talking about cut flowers, are ethylene sensitive. So it is, for our side of the industry, a very significant problem. And I think that's why so much effort has been spent on trying to deal with the problem. And then if you go to the second slide, you'll see that of those stems, we think, or we estimate that about 3/4, actually 77% of the ethylene sensitive stems are being treated today, one way or another. And I think that the result of all this effort to treat these stems has been higher quality and less waste for the consumer, so that they have a better experience at a very low cost per stem treatment.
So Alan, looking at the cut flowers is one way for us to kind of transfer that over the ornamental mark. And Anil, I think you're going to teach us a little bit about ethylene now. But a good way to think about this in the crossover is dianthus. And that's one that's always present in perennials, annuals, and also on cut flowers. So Anil, I'm going to let you take it away now.
Thank you, Bill. And thank you everyone for participating in this webinar. Let's start with the chemistry of ethylene. And it's a very simple hydrocarbon. As you can see in this structure, it is 2 carbon atoms and 4 hydrogen atoms. It's a colorless gas at the ambient temperature, which provides actually certain challenges with this chemistry. And in atmosphere, under normal conditions in open air, the levels of ethylene are very low. They are typically below 10 parts per billion. So under normal circumstances in atmosphere, you don't find that much ethylene in the atmosphere. But later we'll go through some slides and some information that where we will show you where actually we will see more extreme levels of ethylene in the environment. Let's talk about the sources of ethylene. I put them in 2 categories. One source is biological. The other sources are non-biological. With almost any higher plants, mostly flowers and fruit produce large amounts of ethylene, especially when they are maturing as well as when they are ripening. And also, most plant parts produce ethylene under stress conditions. If they are stressed due to water stress or cutting or some chemical damage, plants produce high levels of ethylene under those stress conditions. Also, bacteria and fungi may produce loss of ethylene when they are infected, when they infect either plants or fruits or vegetables. And also there's a non-biological part. Typically it comes with incomplete combustion of organic material. Anything like cigarette smoke or combustible engine heat or exhaust that's a byproduct. Ethylene is a byproduct of that incomplete combustion. And worldwide, ethylene is made for lots of industrial applications. For example, things like plastic. So any pollution due to this industrial production can contribute to ethylene levels in atmosphere. Why are we concerned about ethylene? What is the natural role of ethylene in plants? And it is a plant hormone. That means it is a signal molecule that would affect several plant processes in different stages of development as well as in different species. And since ethylene is a gas, it's very unique compared to other plant hormones. Ethylene is the only hormone that occurs as a gas in the normal environment. And depending on how you look at it, the effects of ethylene could be positive or negative. For example, in growing conditions, for flower inductions of pineapple and bromeliads, you need ethylene, and there are commercial applications of ethylene to induce flower in those species. And also fruit ripening. And in most cases, for example, apples, bananas, tomatoes, ethylene is needed for fruit ripening. There are commercial applications, for example, banana ripening rooms, where ethylene is applied externally to enhance ripening. But unfortunately, especially in post-harvest for ornamentals, the effects of ethylene are not so good. They're actually damaging the quality of plants in post-harvest. So what kind of impact ethylene has on ornamental plants? As you can see in this slide, it induces senescence, also flower and bud drop, in some cases leaf yellowing and leaf drop, and also abnormal flower opening. Sometimes they may not open at all, or sometimes they may be too open. So we see lots of different symptoms with ethylene depending on the species. So if you look at the factors affecting ethylene damage, there are three main factors: concentration, duration, and temperature. Typically, higher the concentration, more severe the damage, and faster the damage. One key thing to understand is the concentration of ethylene that is needed to cause damage is very low. We have seen damage as low as 100 parts per billion, which is 0.1 part per million, can cause. Even lower concentrations, if the concentration persists for longer duration, you would see ethylene damage. So the first one is concentration, then the duration of exposure. Because in later slides, I will show you, how the damage occurs, but exposure for longer time, even lower concentration, could result in damage on plants. Temperature, higher the temperature, more damage, and more easily or more faster the damage would occur. So this here we have in this slide data from a study done in Canada to investigate ethylene levels in different segments of ornamental plant industry. So what they have done is these researchers collected samples of air from different locations and analyzed for the presence of ethylene and the concentration of ethylene. As you can see here, they have sampled air from growers and as well as wholesalers and also retailers. And they were handling different type of commodities, sometimes only fruits, only flowers, sometimes fruits, vegetables, and flowers, a combination of things. And in the last column, as you can see, the concentrations range. There's a wide range of concentrations, but you have, as you can see, in most cases, they had damaging levels of ethylene. And if you can look at this, most extreme cases, there's a combination of fruits, vegetables, and flowers in the same location, which is the case in some supermarket situations or in distribution centers. So what this data shows is ethylene is more prevalent than that we think, and it can occur in different locations, and it's a real problem. So the key thing is to understand in your specialty whether you have ethylene issue, and to understand actually levels of ethylene you are dealing with. Let's talk a little bit about ethylene sensitivity in ornamental plants. Alan mentioned about sensitive and insensitive crops. What we know based on testing of most of the species, ornamental species, some species or some crops are more sensitive than the others. That means some species are damaged by low concentrations compared to the other species. So we categorize them as highly sensitive crops. Even the sensitivity can vary between cultivars or varieties of the same species. And because there's a genetic aspect to ethylene sensitivity, so you would see that difference even within varieties. And also, interestingly, the damage symptoms can vary between species. In one species, you see one damage in the other. So which makes it very difficult to sometimes understand the ethylene damage. Here we have a list. Flowers, bedding plants, and potted plants, what we call highly sensitive species. As you can see, most of the common plant species that we deal with in ornamental industry are sensitive to ethylene. Okay, ethylene damage symptoms. This is a key aspect, I think, in our presentation too. Why I'm saying is the least recognized ethylene symptom, injury symptom, for many reasons. One is what I mentioned earlier. Different species have different symptoms, show different symptoms. The other thing is you may attribute the symptom to some other cause. You may think it is due to a disease or due to a nutrition problem or pest, because there are some common symptoms for all these things. And sometimes the injury can occur maybe a few days after the flowers are exposed to ethylene. So you may not think there's ethylene problem because they were exposed earlier in the chain, but you see the symptom later. And typically, what I recommend is if you see a damage symptom and you can't attribute to any cause that you can think of, most likely it is ethylene damage. So you have to really think about where the conditions of the plants before you got them to get a good idea what those symptoms are. So here I have some photos to show actual ethylene damage symptoms. Here we have orchids. So photos to the left are the symptoms after they were exposed to ethylene, and to the right are the controls where they were not exposed to ethylene. In this case, orchid is a very common ethylene plant damage by ethylene. Yes, you can see the petals can wilt as well as the small buds can wilt. Ultimately, they will drop off from the plant. Impatiens, typically the flower buds fall off, and impatiens is a very common species that we have seen very sensitive to ethylene among the plants. Petunia, we have petunias here. Sometimes the buds stay on the plant, but they do wilt and show some wilting damage. Snapdragon and cyclamen. Again, snapdragons, we saw sometimes the buds fall off, sometimes stay there, and they do get wilting. Cyclamen, either bud or petal drop. A little bit of leaf yellowing sometimes we see. Some indoor plants, as you can see, foliage or leaf drop and also leaf yellowing. Here are some examples of cut flowers, ethylene damage in cut flowers. The first one is carnations. It's a typical ethylene damage symptom that we see, a classical symptom of what we call the petal enrolling. And also in delphiniums, bud drop. Okay, let's have a look at cellular level, how ethylene works. So this is a diagram, just a sketch to show a plant cell. And there are two sources of ethylene to a plant cell. If you look at a plant cell, one which we call the internal ethylene. As I mentioned before, plants themselves can make ethylene inside plant cells. Then we have external ethylene that is coming from the environment, can get into the cell and cause the reactions within the cell. Either whether it's internal or external, it has to bind to a structure called receptor within the cell to start the damage. So the receptor doesn't recognize whether it is internal ethylene or external ethylene because it's the same chemistry. But once it's bound to the receptor, it starts the process of basically the process.
Anil, it basically kills the flower, correct?
Correct. So once in the cellular level, what happens is once it binds the receptor, there's lots of different processes can takes place. Depending on the species, it could at the end result in the bud drop, or the bud may stay there and it may wilt. So the key thing is, the important things for these reactions to occur, the receptor has to be open and ethylene has to go and bind to the receptor. So there are, depending on the species, you would see different symptoms. This is same for even what we are talking about more of the negative effects of ethylene. Even for positive effect, that has to happen. Okay, so now we know how ethylene works in the cell. Let's look at how we can prevent ethylene damage. So if you go back to the first slide that I mentioned, so now we know what it is. Then if we go to the next slide here, we can think of how we can prevent the ethylene damage. I can think of 3 ways of doing it. The first one is basically getting rid of external ethylene. How do we do that? External ethylene in the environment, so that the plant doesn't see that external ethylene. You can do it by either air exchange or sending them to a scrubber or filter to basically get rid of ethylene. That's one way of doing it. Sometimes it's successful, sometimes not, depending on the type of scrubber material you use. So the second way is controlling or preventing the production of ethylene inside the cell. There are some chemistries in market that you can do basically that function. The third one is blocking the receptor. That means you are, especially with some chemistries, you can block the receptor. Once you do that, even if there's ethylene in the cell, it doesn't see the ethylene, and it can't go on start the reactions of the death process. So we call them ethylene action inhibitors. In my opinion, that is the best source because it can take care of both internal and external ethylene. So commercially, there have been lots of work done on ethylene action inhibitors, and still work going on. There are only 2 commercially available ethylene action inhibitors. One is STS, a based product, and the other one is the active ingredient of Ethylbloc. So let's talk a little bit about Ethylbloc technology. Ethylbloc technology.
Anil,
Yes.
Can I interrupt? Because we have a couple of questions here. Before you dive into that, let's address these real quickly. Ramiro wants to know, he says, we mostly have issues with agave, and when he looked at that list you had of plants, he didn't see agave on the list. Do you have any information about agave and their ethylene sensitivity?
We have, what I showed there is a very small section, and we have a comprehensive listing of different species divided into least sensitive or mildly sensitive and medium and highly sensitive. So anyone who needs that comprehensive list, we can provide. And sometimes not all the species are in that list because still we are sometimes doing testing, and especially crops that are not that much common. So it's a list that we will be adding every now and then. But we can provide a list, so anyone who is interested, it's a more comprehensive list than what I have shown here.
Agave is a new one. I haven't seen that one on the list, but it may be still out there. So looks like we have to work on completing that list.
And whenever there are new crops or species, even varieties come in, we can do some screening, and there are very simple ways of screening for any ethylene sensitivity if we are not sure about the sensitivity. and we have protocols that we have developed to do that screening. Akshay, I was not going to discuss it here, but we do have that type of information available.
Yeah, and I think we'll provide information at the end about how you can get some free testing done of some form.
Correct.
Another question, this one might be for Bill, but whoever wants to tackle it. Sunita wants to know, do florists and wholesalers know about ethylene damages to different flower types? Which is a good question. How prevalent is this information?
Hey, Alan, I'm going to turn that one over to you because we've been working with cut flowers for years, and I know your work is in specializing in that area. So can you answer that question, please?
Yeah, I'd be happy to, Bill.
Hang on, Alan. Sunita just clarified. I meant to ask, do growers let wholesalers know about this? I would argue that growers don't know it either.
No, I think, if I could respond to that, hi, Sunita. I think that in general, most wholesalers that have been around a while are familiar with ethylene and the problems that ethylene can cause in the supply chain. I think the problem is that sometimes, even though they're intellectually aware that ethylene is a risk and that it does affect flowers, and maybe even what the symptoms may be, they tend to think sometimes that they don't see that the effect of the ethylene damage at their level, or they say, well, we keep all the flowers in the boxes and we're moving them through so fast that we won't have a problem in our facility, which may or may not be true. And so I think I would answer the question and say I think they're aware of it, but I think a lot more work needs to be done in that area. I think that a lot of wholesalers, a lot more wholesalers than treat today or take active action, to protect their inventory, should be doing that and probably aren't. So that's an ongoing conversation that we have with our customers, for example, about it.
All right. We had a couple more questions about specific crops, but we'll save those for the end since they weren't on the list. So really, I think the agave question kind of covered that as well. So Anil, why don't you keep on going?
Yes, thanks, Chris. So we were talking about ethylene action inhibitors. So I mentioned about two commercially available chemistries. One is STS, has been in the industry for a long time, especially for cut flowers. And Ethylbloc technology is about now, I think, about 20 years old. This is again ethylene action inhibitor. The active ingredient, the chemistry is 1-methylcyclopropene, and you may also have seen the abbreviation 1-MCP. And what's very interesting about this is also a gas at room temperature. So it provides certain unique application methods, as well as also it provides some unique challenges. Although it is a gas, the product that we are selling is the Ethylbloc. What we have done is getting that gas into a powder, and once we want to activate and release the powder, you have to drop that powder into a buffer or liquid. Then it releases the active ingredient as a gas. Several key features of Ethylbloc. It's effective at very low concentrations, typically less than 1 part per million. And it is, from a human toxicity point of view, it's a non-toxic. It's a category 5 EPA category level, which is very benign, non-toxic chemical. And there's no special disposal requirements because of the low hazardous nature of the product. And one thing about the plants is a very important thing is if you overdose accidentally or for some reason, it does not harm plants. You can't say that about many chemistries. Even if you, let's say, 10 times overdose it, we haven't seen any negative effects. It may not give any additional benefit, but the key thing is if you, for some reason, accidentally overexposure, they are not going to harm plants. Here's a small diagram, going to a little bit detail into cell. to show how 1-MCP or Ethylbloc active ingredient works. It's basically what it does is blocking the receptor I mentioned about you a few slides earlier. So if you look at the receptor, it's typically bound to the cell membrane, and there's a place called copper center there, as you can see in purple. And once ethylene goes and binds to the receptor, as you can see in the second diagram, it opens up these arms of this receptor. That's where the response happens. So what happens is if you treat the cell with 1-MCP, it goes and binds to that place where ethylene goes and binds, basically blocking that space so that ethylene can't go there and bind. So even if ethylene is there, there's no response. That means no damage done. Ethylbloc technology, there are many forms that's available for depending on the country. We have what you call the truck kit. It's designed for larger volumes like a container. They come in two different sizes for a smaller truck container kit or bigger one. And there are some information about the size of the product and temperatures. And also it comes as a tab sold in Europe. It's again the same technology. They come in different forms. With the tab, there's actually a kit that will be provided to activate the active ingredient. So this series of photos show how to use the Ethylbloc truck kit. So so that kit is designed for certain volume, as I said, or bigger truck container or smaller one. So how you use this is once the loading of the truck is done, whether it is cut flowers or potted plants or anything done. So this is the last thing you do just before closing the door of the truck container. So basically to activate it, it comes, there's a buffer that's in a container, and also the powder of Ethylbloc in a water-soluble pouch. So you pour in the buffer into the container and then drop the water-soluble pouch inside, and it dissolves and start releasing the 1-MCP active ingredient gas inside the truck. So once you drop that pouch in the container, you should close the door and leave it closed at least for 4 hours for the gas to release and work through the whole container. Since it's a gas, it will diffuse very fast from one end to the other and give the right concentration within that truck container.
Hey, Anil, I wanted you to talk about this a little bit, how it dissipates in the container. But one thing we didn't touch on is how fast ethylene can damage crops. And I know a lot of people on the line here load their trucks the night before. They're going different distances. So how fast can ethylene damage occur whenever you're starting to ship products?
So, Bill, what you mean is once plants are exposed to external ethylene, how fast you can see the symptom?
Or how fast is it 1 hour, 15 minutes, 2 hours after being exposed?
In our experiments, sometimes we do different concentrations and see how fast you can see the symptom. And we have seen even within 15 minutes, if the concentration is high, you would see the damage. And in some cases, let's say the plants are exposed to ethylene, the symptom may occur later. You may not recognize it because in some processes it takes time because there are cascades of different things going on. But we have seen from like 15 minutes to days. So it depends on the species, but it could be very fast. So the important thing is, what my recommendation is, treat with Ethylbloc as early as possible, so that what happens is once the molecule of one MCP binds to the receptor. It stays there for some time, sometimes up to 10, 14 days. So once you treat, the flower is or plant is protected. Although ethylene, you see ethylene, once the flower is protected, you don't get the damage. So for that reason, it's much better to apply the treatment as early as possible, especially if when you're shipping, apply it just before shipping, so that, because especially during shipping and when it is lots of volume of crop in a smaller area, the buildup of ethylene is very high. That's where you need the protection.
Okay, thank you, Anil. So
the second form of Ethylbloc technology we have, it's a sachet, a small packet. It has 5 grams of powder in there. And this is designed for small boxes. It could be a cut flower box, it could be a box with cuttings, or a box with a potted plant. So in this application, you dip a sachet in water for just a second to activate and drop it in the box and close it. Then start releasing the 1-MCP and start acting, giving the protection.
and this is used quite a bit on most orchids. In fact, the orchid market has changed tremendously because of the ability to take and treat orchids in the individual boxes. So
Sure.
the sachets are a huge protection for the orchids today, especially Phalaenopsis.
Anil, how long does a treatment last? One of our attendees wants to know if there's any way to protect a plant from basically like from seedling to fully grown and shipped out to retail.
Well, typically once remember the way the mode of action is that 1-MCP molecule goes and binds to the receptor. After some time, that molecule is going to get away from that receptor. Typically, it is about 10 to 14 days. So if you apply at seeding level, could you get the effect after 2-3 months? No. But if you're applying for controlling certain things, let's say if you want to mitigate the stress level, there may be indirect effects of what you have seen at that level, but the molecule may not stay at the same receptor pool. Typically, it is about 2 weeks, 2-3 weeks maximum.
Okay.
Now, again, new receptors are formed when there are new organs formed. So you had to retreat to make sure those new receptors are protected.
So it doesn't sort of spread through the plant. It lands on receptors. As the plant grows, gets more receptors. Those receptors are not protected.
Correct.
Gotcha. Okay, thanks.
Let's go into some photos showing the Phalaenopsis orchids. It's very common effect of Ethylbloc. As you can see, it is protected by Ethylbloc treatment. Over here, you can see the negative effects of ethylene damage is protected by Ethylbloc. As you can see, different crops show different symptoms, but typically most of the effects you can see. And I have lots of examples of different crops, but I'm not going to share lots. And one of the things that I should say that is probably 1-MCP is the most researched chemical for a long time. Lots of academic research available to look at the rates and the damage and everything. So it's lots of information out in the public domain. That's it for my section. Bill, I think it's time for Vijay to talk about cuttings.
Yeah, it's time for Vijay, and this is something that we've started a lot of work on recently, and Vijay's heading this up here, and the effects of ethylene gas on vegetative cuttings. All of us are growing lantanas today. There's a lot of these new euphorbias in the market, poinsettias. All of these plants are susceptible to damage from ethylene gas. So Vijay's going to talk a little bit about his work, and then I'll turn over to Sanal to talk about some of the other things we're seeing in the market too. So Vijay, why don't you take it away?
Thank you, Bill. So Anil talked about the effects of ethylene and Ethylbloc mode of action. So I think that's all cleared up. So I don't have to talk any about this mode of action related things. Just I want to go directly address the applications in the unrooted cuttings. So I want to talk quickly about the vegetative cuttings, the supply chain, and when you're producing plants in the Europe and in North America, most of these the material for starting these plants are coming from Central America, East Africa, China, and Israel, and so on and so forth. And and the plant material is extremely sensitive, need to be shipped and initiated. The propagation needs to be initiated within 72 to 96 hours. And during this time period, there are a series of events that are happening. Actually, cuttings go through a roller coaster of events. The first thing that is going to happen is the harvest. And immediately after the harvest, the temperature will be brought down using passive cooling or forced air cooling, and they will be boxed, they will be trucked, they will be on the airplanes, and then they will be again on the trucks before the propagation is initiated. The industry does a really, really good job managing the temperature, the cold chain management during the shipment, and it is really important to cut down the respiration rate. But one other thing that is really kind of being not actively looked at is the management of ethylene, because immediately after you harvest, there is a wound ethylene that is being produced. If you're chilling the cuttings, the chilling ethylene is being produced. Vibration during shipment, ethylene is being produced, and so on. So some of the ethylene sensitive crops show these symptoms once you receive the cuttings. Next slide, please.
And one other thing, Vijay, you and I have talked about the transport of the cuttings too. Even vibration in transport can cause problems in creating extra ethylene in that transport. And that goes for cuttings and also finished crops too, correct?
Yeah, that is correct. So talking about some of the commonly seen issues in the industry, poinsettias, typically you see leaf senescence along the midrib, but before the leaves turn fully yellow, you have leaf abscission. Euphorbia hybrids, also the same thing. You have severe leaf loss. Portulaca, the same thing. Lantana, it is a little bit more complicated. Actually, we'll talk a little bit more about lantana in the next slides. And the last one is geraniums. You have severe post-harvest leaf senescence in geraniums, and ethylene is partially involved, but it's not all about ethylene. There are other things that are causing the yellowing in geraniums. Okay, so here is the leaf abscission in Portulaca. Before I actually walk you through the slide, let me clarify one thing. There is an interrelationship between ethylene and the carbohydrate status of the cuttings. So what does that mean is the more the carbohydrates, more the sugars, more energy in the cuttings, they're less sensitive to ethylene. And the less sugars, the less energy, they're more sensitive to ethylene. So if you look at the graph here on the left side, on the x-axis, you can see the various times of harvest. The cuttings are harvested at 8 a.m., 12 p.m., 4 p.m., and so on. And on the y-axis, you can see the number of leaves abscised per cutting. Okay, and without Ethylbloc, which is the red line, you can see that there is a significant leaf loss that happened at the 8 a.m. in the morning, and whereas significant less leaf drop that is happening when it is harvested in the evening. However, when you treat the cuttings after immediately they're harvested, there is no difference between the time of harvest. 8 a.m., 12 p.m., and 4 p.m., there's a similar, there's no difference, and you can significantly inhibit the leaf abscission in Portulaca. Okay? Talking about lantana, actually the problem is a little bit different here. This is actually, I would say, this is the worst among the bunch. Leaf abscission is a big issue, but also not only leaf abscission, there is also necrotic spotting is also a big issue with lantana. Changing the time of harvest or applying Ethylbloc. Of course, applying Ethylbloc is much more, you can rely on that. Applying Ethylbloc can eliminate or reduce significantly the abscission of the leaves. But if you're having certain cultivars that are sensitive to necrotic tips, what you need to do is you need to reschedule the time of harvest of those cuttings. Harvesting towards later part of the day will help with avoiding the necrotic tip formation in lantana. And also with lantana, you want to avoid the mechanical damage while you're harvesting and handling the cutting. The damage while handling the cutting should be minimal, because this is one crop that is sensitive to mechanical damage also. So both by applying Ethylbloc and rescheduling the time of harvest, if there is a sensitive cultivar to necrotic spotting, you can control both issues in lantana. Okay. I just want to walk you here with poinsettias. Again, poinsettia is also very sensitive to leaf abscission. On the left side, there's a graph. On the x-axis, you can see without Ethylbloc. On the right side, which is treated with Ethylbloc. When it is not treated with Ethylbloc, there's a significant leaf abscission that is happening in the untreated cuttings, and there's a significant less leaf drop that is happening when it is treated with Ethylbloc. And you can also see the rooting response on the right side, control on the top, and then the Ethylbloc treated ones on the right side. Ethylbloc application doesn't affect rooting response in poinsettias or most of the crops. Geraniums is one, might be delayed by a day or two, but it is not significant compared to the damage benefit you get from Ethylbloc treatment.
A question about treatment in boxes. Hank wants to know if the box has vent holes in it, will the Ethylbloc sachet still work? Because it says to seal up the box, and of course, every cutting box I've ever seen, I think, tends to have holes.
Yeah, great question, Chris. Actually, I'm going to talk about that in the next slide.
Oh, I should have paid attention.
No problem. All right.
There we go.
So yeah, so here, just wanted to talk quickly. Recap the list of crops that are sensitive to ethylene. These are already well established. We have done research our last 10 years. A lot of the groundbreaking research was done 10 years ago related to the time of harvest and so on. But here are the already established list of crops that are sensitive to ethylene. There might be other crops. If you are seeing leaf abscission or leaf yellowing, it's really worth looking at that. But if you go on look at the list, poinsettias, lantanas, portulacas, euphorbia hybrids, Thunbergia, agastache, and geraniums, I put it in parentheses as a partial benefit. The reason is, again, geraniums, actually I spent most of my early adulthood studying about geraniums and the relation between carbohydrates and leaf senescence and so on. That is a little bit more complicated. You only get a partial benefit using Ethylbloc because beyond ethylene, there are other responses that are causing leaf yellowing in geraniums. So I would like to, because of the time, I would like to hold off on that. We can have some other time do a webinar on that. But that's the list of the crops. Talking about the application, Anil already talked about the method of application. I'm not going to cover all that, but just want to touch on a couple of important things. The sooner the better. The sooner the better. Right after you harvest, again, as I said, immediately after harvest, you have wound ethylene, chilling ethylene, and all that stuff that is happening. So the sooner the better, immediately you can apply Ethylbloc, that is better. And especially when you are dealing with unrooted cuttings, again, these cuttings, depending on the type of the cuttings, these are packed in small bags. If you are packing in small bags, make sure those bags have perforations and use an outer bag so that these small bags can be packed inside that outer bag and place an Ethylbloc sachet inside that bag. So that should help. Again, that outer bag will minimize the escape of the ethylene and Ethylbloc, and you should get good treatment with the MCP.
Okay. And Vijay, Annabelle was wondering about the number of sachets per box. She couldn't find any instructions in her Ethylbloc.
Sure.
Based on the size of the box.
Yeah, actually it is because the cuttings are really packed in high densities and so on. It is typically the recommendation is 2 sachets per 3 cubic foot bag, but we can have those information available. We can send that information. Okay?
All right, very good.
Okay, I'm handing off to Sanal.
So one of the things that Sanal is going to touch on here is that there's a lot of other ways that are being perceived to help reduce ethylene damage. And one of them is calcium chloride spray. And we did some work here, and Sanal actually did a study on petunias, and he's going to cover calcium chloride and its role in protecting crops during the supply chain. And this is a big part of protecting crops in the insurance. He's going to show you some of the results, and then I'll take it over after that. So Sanal, take it away.
Thank you, Bill. Appreciate it. So today I'll be talking about a study we did regarding calcium chloride and Ethylbloc. It was done with Petunia, Supertunia, Royal Velvet from Proven Winners. So let me give a background of why we did this study. A common industry problem involves botrytis infection of bedding plants during post-harvest shipping. So the most common example is the meltdown of Petunia flowers. So calcium chloride has been reported to reduce flower meltdown caused by botrytis cinerea during transport and storage. So there are questions in the industry whether treating with calcium chloride could also alleviate ethylene damage during transport and storage. So with this in mind, we did a study whether we wanted to understand whether calcium chloride did prevent ethylene damage. So we got a commercially grown petunia plants, Supertunia Royal Velvet, at full bloom, purchased from a local greenhouse. And these three treatments were applied to the plants. In the first treatment, here we can see potted plants, petunia plants were challenged with ethylene. And then the next treatment is the petunia plants were treated with calcium chloride at 1,200 ppm. And then it was challenged with ethylene. In our third treatment, the petunia plants were treated with Ethylbloc and then challenged with ethylene. So here we used Floral commercial formulation to generate around 2 ppm ethylene to induce the ethylene damage. Next slide, please. So coming to the results, here we can clearly see that the potted petunia plants 4 days after treatment, we can see in this picture that flower drop is clearly visible due to ethylene damage in treatment 1 and 2. In treatment 1, it was just the ethylene at 2 ppm, and in the treatment 2, calcium chloride 1,200 ppm, and then challenged with ethylene. Both we saw ethylene damage. In the third treatment, Ethylbloc was treated, and then ethylene challenge was applied around 2 ppm, and there was no flower damage that is clearly visible. So this study concludes that calcium chloride application will not reduce or suppress ethylene damage. And we can confidently say that calcium chloride cannot substitute Ethylbloc technology for preventing ethylene damage. So with this, I will hand over to Bill.
Thank you, Sanal. And I think one of the things about the calcium chloride, it does help protect the plants from botrytis. It toughens up the cell walls and the membrane. So if you're really looking to protect your crop, this is just another part of insurance. And this is kind of the last question. How are you planning to manage your risk during the supply chain? And going back to unrooted cuttings, Vijay talked about that and how far they're transported. We're very lucky that most of the breeders in the market today are using some type of strategy in insurance to make sure you get good cuttings. There's a lot of ethylene is everywhere in the transportation supply chain. So while you may not have a problem, it could be worth taking and buying a little bit of insurance. We worked hard to get this far. It's worth the investment to look at your facilities, how far you're transporting product. And one of the things that we can provide for you, and Chris, you want to head to the next slide, is that we can develop a risk assessment. We do this with cut flowers, but we've developed one for potted plants. And it's just some basic questions, and we can go through those questions, and there may be a chance that you don't have a problem. But what we find is, even if you're only transporting maybe for 50 miles or 100 miles, you may pack the trucks the night before, and they may sit there for 5, 6, 7 hours so that it's ready to go the next morning. You're at risk. So like Anil said, it could be 15 minutes or an hour. You could already start to see that damage. So if you go to the oasisgrowersolutions.com/risk, you can sign up there, and we can contact you, and we can fill out a risk assessment. We also, if you are at risk, we do have sample kits where we can measure the actual ethylene gas levels in your facility, coolers, or trucks. So that's a big part of it. That's really all I've got today, Chris. I know we're a little bit over, but I'm going to turn it over to you, I guess.
All right. Well, let's take a few minutes for a few of the questions that have come in, or if anybody's got any new ones. Let's see, let me scroll back a bit up here. Wanted to know if it's labeled, Bill wanted to know if it's labeled for vegetable transplants, or I would ask, are there any vegetable transplants it's necessary on if it is labeled?
Well, that's an interesting question. There is a product out there in the United States now that is labeled for vegetable transplants. It's called Landspring, and it's mainly been a lot of work done on field tomatoes, and it does reduce the stress of transplant, and they see a good improvement in yield going through the supply chain. So there has been work done. It's a little bit different application and methodology to it, but there are benefits to seedlings, especially tomatoes, using the 1-MCP technology.
All right, here's an interesting question from Eric. Have you done any studies, this applies to trailer treatments, any studies to see if the distribution of the gas in a trailer is reduced or impacted by the vacuum of a truck going down the road?
I don't know about the vacuum, but as long as the truck is sealed whenever it's applied, it takes and dissipates into the trailer pretty quickly. There is probably one of the most limiting factors during the application is standpoint of temperature. And Anil, I think you're on. If you can kind of talk a little bit about that, but I've never heard about the vacuum, but maybe you can help us out there, Anil.
Yeah, Bill, although we haven't done actual measurement of 1-MCP inside the truck trailers, but we have done some studies where we put test samples at different locations of a truck and send it through a typical Ethylbloc truck treatment kit, and we have tested plants or flowers. They were located from the furthest end and the closest end, and we have seen a good distribution is there. And yes, there may be a little bit of reduction of or loss of ethylene during any of the exchange and things like that, but there's enough material there in that truck kit to cover that type of losses. So yeah, we have done some studies to make sure that the distribution is inside the truck is valid.
What I suggest, Anil, next time you're thinking of one, try it doing about 70 miles an hour down the highway in a trailer, and we'll get a firm answer on that one.
So maybe you can drive the truck.
That sounds like a deal. Bill wants to know, and I don't know if you'll have an answer to this one or not, but let's give you a try. Bill wants to know, are there any figures for concentration of ethylene produced by internal combustion engines from vehicles that might be running around a greenhouse or a nursery, tractors, forklifts, etc.?
Probably I can answer. I don't have any specific numbers. It depends on the type of combustion if it is incomplete. So there may be numbers available. Remember, it is where the that ethylene production takes place and how fast it dissipates the area and all these things. So it probably is very difficult to figure out a localized volume, but more trucks and more things in a contained environment, more ethylene you would expect to get.
And Chris, can I add to that a little bit? And this is anecdotal. I have been in a lot of greenhouses where you're running the forklifts down the aisles, and I've seen New Guinean patients show signs of ethylene damage from the forklifts running down the aisle. So it may not be a closed environment, but when you are gassing them right along that, I have seen that damage in a greenhouse.
Yes, and that applies to a question Jennifer brought up. She's a Kalanchoe grower, and they've got construction going on with welding taking place in the greenhouse, and they say they've been seeing some flower damage typical of ethylene, but they're seeing some other types of symptoms as well. And can ethylene cause plants to revert to a vegetative state?
Anil, I guess I answered that one. That one.
I'm not sure. I do
Let me finish the question, Anil. She says she has some plants that would typically have 15 stems with 15 flower clusters, but we're seeing only the top 4 flower clusters form properly, and then the side shoots have no flowers.
Yeah, that is possible, although it is probably not revert. But we do know that certain species need ethylene for flower induction. Now, once the flower is induced, whether it will go back into revert into vegetative state, in some cases may, but I am not 100% sure in what species. But the thing is, sometimes with ethylene, remember it's a hormone. Typically with hormones, there are interactions with other hormones, so it's a complex situation. So you can't just take ethylene itself and make a decision of what's happening in the plants. It's sometimes it may interact with some other hormones and does something unusual. Yeah, what crop are we referring to here?
Kalanchoes.
Kalanchoe.
Which is highly sensitive to ethylene. So depending on the amount of work in the greenhouse, it could be one of the causal agents, but that one's a new one. I don't know how much ethylene gas is produced when you're welding.
All right.
And as you know, ethylene, like floral or ethereal, used in lots of crops to keep the flowers short. So sometimes it slows down the flower induction in certain cases. So it's a complex situation depending on the crop.
All right. One more here. We've got Ali is a buyer, I'm assuming a flower buyer, and would like to let his vendors know about the product. Is there a more condensed version of this presentation that I could pass along? I imagine you have some sales material that could help Ali with the people he buys his flowers from.
Yes, we do. Yeah, go ahead. Sorry, Alan.
We have lots of information. You can go to the Floralife website or the Oasis Grower Solutions website, download that, or if you sign up on the oasisgrowersolutions.com risk assessment, we can get you all that information and follow up with you. But it is available online.
And there's that address one more time. Jot that down, everybody. And I'll tell you what, if you've got, there are some more questions, some crop-specific questions involving, well, I saw bougainvillea in there. We already talked about agave, a few others. Shoot those questions directly to my co-host, Bill, at Riffy, that's for his formal name, William, which only his mom calls him, at Smithers-Oasis.com. And he'll direct the question out to the appropriate scientist out there to help you out. It's pretty technical and a lot of us, very specific issues, but I know these guys, obviously they know their stuff and can help out. So, sorry if we weren't able to get directly to your question, but we'll get them answered one way or another. And if you want to relive this webinar, go back into some of the fine points or share it with colleagues or your vendors or buyers or whoever it may be. You will be able to do that within the next, certainly within the next hour or so at growertalks.com/webinars, conveniently the same place you sign up for these webinars. And so with that, we're going to wrap it up for Vijay, for Anil, Sanal, Alan, Bill, and all the fine folks at Smithers-Oasis, and for all my peeps at Ball Publishing who work so hard, really so I don't have to. This is Chris Beatty saying, so long, everybody.